So let's close out this for now with this paper, which actually focuses on a broader aspect of the role of biology in reducing greenhouse gases in the atmosphere. So it's not just agriculture, it includes other ecosystems as well, as we will read. I'll just grab some points they made and read them off. Again, you have to follow up with the vast literature they provide as references if you're interested in this direction. To begin exploring possibilities, a group of researchers with expertise spanning synthetic and systems biology, atmospheric and terrestrial science, policy and ethics met in Boston in 2019, so not too long ago, to discuss some of the potential applications of SSB and its impacts on sustainability and atmospheric carbon drawdown, and to address the technical and social challenges of large-scale implementation. So it's along the themes we have discussed for adaptation, but focused much more on biology, especially synthetic and systems biology. They have definitions of what qualifies as synthetic biology, and systems biology is also a broad framework kind of descriptor that you can look up if you're interested. Several interrelated themes emerged, including the following. There is a potential role that SSB might play in diminishing climate change and its impacts by contributing to regional and global sustainability, which is great good news, drawing down atmospheric carbon or reducing emissions, which is part of the mitigation as well, avoiding the release of CO2 or methane into the atmosphere. So that we have discussed a little bit about cattle and chicken and land sparing and rice. Reducing water use in rice can reduce methane emissions, right? The wide range and timescales that would be needed to for proof of principle, scaling and cost reductions depends on the specific application. The economic cost benefit of SSB, which would weigh heavily in favor of benefit as a consequence of substantial co-benefits, including less expensive, more effective crop growth, which we discussed a little, the ability to use degraded lands that are currently not sufficiently arable for the growth of crops and other useful plant products, the potential for discovery of plant-based pharmaceuticals and biomaterials, and more generally a boost in the growth of agrogenomic startups. Okay, so these are generic points, but general points rather, but still useful as co-benefits. And as for using degraded lands, this comes up in many ways, saying this is what can serve for bioenergy, but it is yet to be shown at scale in terms of actually growing efficient bioenergy species on degraded lands. An emerging roadmap for future research, which includes an open and global assessment of serious ethical, social, legal, environmental and scientific issues that must be resolved before SSB can be introduced as a climate control measure. So even though I'm putting this in adaptation, this is obviously a intersection of adaptation and mitigation. So it's talking about many adaptations for reducing greenhouse gas emissions. Further, we need much deeper understanding of managed ecosystem dynamics and how it impacts and is impacted upon by climate if we are to evaluate the potential effects of any mitigation strategy. One example is the coupling of nitrogen and phosphorus cycles in land dynamics. So if you tinker with one, you change species composition, for example, especially in coastal waters as well. An inadequate allowance for the linkage could lead to failure when trying to introduce engineered organisms with nitrogen fixing capabilities into natural environments. Basically, most vegetation and photosynthesizers take nitrogen in a reduced form, ammonium especially, but there are some who can directly do dinitrogen into nutrients in their cells, so they are nitrogen fixers like diazotrophs. Similarly, the impact of rapid CO2 drawdown on the symbiotic assimilation association between the multiple types of mycorrhizae fungi and the plant roots with which they associate. There is a symbiotic relation required for nutrient uptake, etc. and which play a critical role in carbon assimilation, involves a complex dynamic that will require extensive experimental and computational research to achieve a reasonably predictive understanding. So this is basically to avoid Frankenstein consequences, Frankenstein experiments with unintended consequences. Decisions concerning the development and implementation of technologies that biologically manipulate ecosystems should be made on the basis of a careful assessment and evaluation of the benefits and risks of different options. This will require time and extensive experimentation, as it's extremely difficult to model even small ecosystems accurately enough to understand perturbations. This is a good, humble statement, because lots of dynamic vegetation models and the crop models we looked at actually can be not as clear in saying what the limitations are. Okay, we need a much better understanding of biological engineering at very large scales involving communities of organisms. What can be engineered to work in a microbe in a petri dish may not work if scaled up to a bioreactor or an industrial fermenter. So biosynthesized enzymes and various materials are being used already for stabilizing land, etc. So you can follow up on the exciting literature there. Some progress has been made in identifying and manipulating small communities of organisms that enable a particular environmental target to be achieved, such as nitrate reduction, carbon sequestration, or heavy metal contamination. So it's the idea of dealing with ecosystems than individuals. Rather than dealing with just nitrogen fixers, can you look at the biome in which, or the ecosystem in which they live, and you deal with communities of organisms than individual organisms, and then see if ecosystem works better in that sense after the experiment or after the perturbation or biological engineering. The experiences and expertise of the industrial and biopharmaceutical communities will be valuable and necessary here if SSB engineered approaches are to be used. Unlike sugar fermentations that are typically carried out in batches, fermentation of carbon dioxide or methane is typically carried out in a continuous fashion. Yet outside the biopharmaceutical industry, the impacts of continuous fermentation for manufacturing are not well understood or modeled. Okay, some details, but they are critical for doing large-scale SSB engineering. The development of improved methods for containing, replicating engineered plants and organisms must continue to receive high priority. Okay, the cost-benefit ratio, which varies with the type of technology, needs to be carefully analyzed, since it will be central to any policy decisions. To read off some more, it's important to note that the US and some other countries like China and some African countries are more accepting and tolerant of genetically modified organisms than Europe. This underscores how important it will be to get the ethical, legal, and social implications, called ELSI, and other societal acceptance efforts right. Europe in general is more aware, more progressive in many ways, and maybe less accepting of unknowns in that sense, like GMOs. Climate change is a global challenge requiring international efforts to determine how to work together effectively and harmonize different approaches. The scale of what is contemplated in terms of negative emissions by manipulating cultivars and forests is huge. For this mission to be successful, the Convention of Biological Diversity will have to be respected, as well as its Cartagena and Nagoya protocols have to be respected, which are under the UN aegis. The ELSI-related R&D can proceed in parallel with mitigation R&D so as to neutralize potential negative parts of the mitigation strategy and confirm the safety of the rest. Okay. There are many other scientific challenges and opportunities that were mentioned and will be fully discussed at the next international workshop, which I will record later if it yields new information. These include the following: the possible development and application of SSB for improved photosynthetic efficiencies and increased atmospheric CO2 capture, so kind of a carbon capture and utilization in a biologically engineered system. Consequences for an organism or an ecosystem if an organism had a better form of photosynthesis. So what does this mean when we improve photosynthesis? The possibility and desirability of safely introducing photosynthesis into currently non-photosynthetic organisms. So there are heterotrophs. Can we tinker with their ability to maybe photosynthesize and make them mixotrophs? Elsewhere I have recorded a podcast on how mixotrophs are present and have dual strategy of photosynthesizing and also ingesting particulate matter and enhance carbon uptake in the ocean. Okay. The possible alteration or adjustment of biomass distribution in multicellular plants, potentially increasing productivity, which we looked at as yield in genetic editing approaches. The possible use of SSB technologies to redirect greater amounts of carbon into subsurface partitions. Coming closer, the possibility of engineering organisms in addition to E. coli to use CO2 as a primary energy source so that CO2 uptake is enhanced naturally. Are these nature-based solutions if we engineer them? Will our engineered biology, synthetic biology, become part of the nature? Critical questions. Possible use of SSB technologies to increase methanotrophy, which is the consumption of methane. Methanogens methanotrophy are important over the evolutionary timescales, but now since we are increasing methane production and concentrations in the atmosphere, we have to worry about such SSB technologies as part of the adaptation and mitigation. And methane is 25 times as strong greenhouse gas as carbon dioxide, even though its lifetime is only about a decade compared to carbon dioxide, which has got a residence time of centuries. So all these things matter. Concentration, residence time, and global warming potential matter. The development of biomes with controlled conditions that will enable assessment of SSB intervention strategies has to be part of the agenda as well. In addition, a number of very important legal societal questions need to be addressed, such as the following. How can we best prepare for unintended consequences of intervention? Frankenstein experiments are always a possibility when we tinker with these kind of biological and other systems, and pretty much everything we have done has had unintended consequences, from agriculture, from the end of the Ice Age to Industrial Revolution. One can argue lives have gotten better, lifespan has gotten longer, but now we have global warming. So how can we alleviate global warming, or will alleviation of global warming create another side effect that we are not foreseeing? Who is liable if an application of SSB technologies harms someone or some community? This applies in general to all geoengineering type of experiments which are being tried, like enhancing alkalinity in the ocean to increase carbon uptake, or spraying aerosols into the stratosphere to do solar dimming, because we know volcanic eruptions, which does solar dimming, naturally has unintended consequences in terms of droughts, crop losses, and so on. Will the hoped for benefits be fairly distributed? Who will win and who will lose? These are, of course, very critical questions. Okay, so here is an infographic from the same paper. So you can see here the role of synthetic biology in atmospheric greenhouse gas reduction, prospects and challenges. So this is from this convention here, Biodesign Research. Genetic engineering as a tool to fight climate change. Decreasing atmospheric carbon dioxide, major challenge in addressing climate change. Synthetic biology and systems biology, genetic modification of biological systems to increase CO2 removal. Before we looked at food security, crop yields, crop resilience to disease, drought, etc. Here we are looking more specifically at greenhouse gas reductions. Powerful tool against climate change crisis if it has no dangerous unintended side effects. So with international cooperation, SSB can be a valuable part of an organized strategy to mitigate climate change. Potential applications, which we read off: altering root to shoot biomass ratio to increase carbon sequestration, increasing agricultural productivity to decrease land area needed for high yield, which also we talked about in terms of genetic editing, which is not independent of this SSB portfolio. Adding nitrogen fixation properties to non-legumes to decrease nitrous oxide production. So evolutionarily there are nitrogen fixers, but can we add genetic engineering to make non-legumes to become nitrogen fixers as well? Modifying fast-growing organisms like algae and bacteria to use CO2 as a fuel. But then again, if they start multiplying and go crazy, what happens? Who pays the price? Challenges in SSB then: assessment of unknown risks, education and outreach to increase public acceptance, and practical applicability, scalability, cost-benefit ratios, etc. Okay, so this is just a handful of podcasts to give you a sense that adaptation to climate change is not just about infrastructure, resilience, and early warnings, but also using these approaches for sectors like food and biology in general. But you can do this also for transportation, water, energy, health, and so on, which I will add, as I said, if I get good literature or books. Okay.